To David 7 Science Theatre, Uganda. Today we are going to talk about insulin. Yeah, you enjoy the video. That is David 7 Science Theatre. And we have a lot concerning science, just like us. Don't forget to subscribe for more and better videos. There are many on our page. So you subscribe on our channel, you'll see them there. Very many science videos. A lot of teachers, a lot of people, a lot of knowledge, a lot of wisdom. A share to other students to also enjoy. Today, as we said, we are going to talk about insulin. So we are starting with the secretory unit in the pancreas. The secretory unit has the acini and also has the islet of Langham. These four types of cells at the center. You also have blood capillaries. Yes. Now, the four cell types in the islet of Langham is called, we have the alpha cells which secrete glucagon. We have delta cells which are secreting the somatostatin of pancreas. We have beta cells which will secrete our insulin we're going to look at today. And we have the PP cells which secrete pancreatic polypeptide. So that is the secretory unit within the pancreas. These red structures are just blood vessels, call them capillaries. How are the four cells of the islet of Langhans connected? Yes, first, the beta cells will release insulin when glucose levels rise in blood. This insulin will stimulate production of glucagon by alpha cells. By this, glucagon stimulates both release of insulin and release of somatostatin. Somatostatin being released from delta cells. Now, the released somatostatin will inhibit any hormonal release within the pancreas. So it means to inhibit release of pancreatic polypeptide, inhibit release of insulin, and inhibit release of glucagon. So it inhibits all the four cell types. Yes. Let's look at this. Look at this. When insulin is being formed, it is formed from pre-proinsulin, which has 108 amino acids. Then it goes to proinsulin, which has 86 amino acids, and final insulin, which has 51 amino acids. So what occurs? To get in pre-proinsulin, we have the signal sequence attached to the proinsulin. So it means in the endoplasmic reticulum, they cleave off the signal sequence. Now the next stage occurs in the Golgi, where the formed proinsulin is now cleaved off the cis peptide. This is important clinical wise in checking whether it is type 1 or type 2 diabetes. And if it is in the type where there is no formation of insulin, it means the C-peptide won't be there. But if there is formation of insulin and you have diabetes, the C-peptide will be high in concentration. So the formed insulin has 51 amino acids, but on carbon 7 in the B chain and carbon 7 in the on residue 7 in the B chain and residue 7 of the A chain, we have a disulfide bridge. We also have a disulfide bridge between the 19 of beta chain and 20 of A chains. Yeah. So that is insulin. Cleave off signal sequence, cleave off the C-peptide, and you form insulin. Let's continue. Biphasic response in insulin secretion. When does this occur? You see that curve? Yes, it has two phases. One phase of a peak and another phase of gradual increase. So when does it occur? It occurs in response to continuous glucose stimulation. Yeah, if you continuously supply glucose, that biphasic occurs. The first peak is due to release of preformed insulin from the vesicles. When it gets depleted, we see it decreasing. So now the next phase, the second phase, is due to glucose stimulation of insulin synthesis that sustains the secretory phase. So now the glucose is stimulating formation of insulin to sustain the secretion of insulin. Yeah, that is the biphasic curve. Role of nutrients in insulin secretion. Exogenous nutrients. These ones will link the nutrients in insulin secretion. The exogenous nutrients, we have glucose, amino acids, fatty acids, we have keto acids, we also have potassium and keto acids. These ones will stimulate secretion of insulin. When insulin increases, it increases or stimulates uptake and metabolism of these exogenous nutrients. Yes. Now we have this curve. When you look at this curve, there is plasma glucose and plasma insulin response. Yes, how release of insulin responds to increasing plasma glucose. You see, from 0 to around 50 or 70, we have some like below 50, this glucose concentration cannot stimulate any release of insulin. So when you're heading towards from 100 to around 250 or 60 or 70, around there, you see, to around 300. Yeah, you see there is increasing insulin release in response to increasing plasma glucose. Now beyond 350 and more, you see there is constant release of insulin in response to blood glucose. Glucose in blood stimulates insulin release from beta cells. How? When you have glucose in the arterioles that supply the pancreas, this glucose will now enter the beta cells through the GLUT2 transporters. Now, through that process of glycolysis, the end product we have and citric acid cycle, oxidative phosphorylation. What we shall end up with increasing levels of ATP. When ATP rises, the ATP will block the potassium channel which pumps out potassium from inside the cell to outside. When it inhibits this channel, it means potassium ions rise inside and causes membrane depolarization. The depolarization of the membrane will cause opening of voltage-dependent calcium channels. Calcium will enter the beta cell and will cause fusion of insulin-containing vesicles with the membrane of the beta cell, and insulin will be released by exocytosis. So secretion, that's how it occurs in response to increasing glucose in blood. Okay, let's continue. Role of autonomic nervous system in insulin secretion. Yes. Sympathetic nerves and epinephrine. Yeah, epinephrine is most predominant inhibitor of insulin release. In emergency situations like stress, extreme exercise, or trauma, the nervous system stimulates the adrenomedulla to release that hormone. Epinephrine suppresses insulin release and promotes energy-yielding compounds like glucose from the liver and fatty acids from adipose tissue. You get? Now, parasympathetic nerves to pancreas and the acetylcholine released will increase insulin secretion to some extent and stimulate insulin release. You get? Okay, let's continue. Half-life of insulin. This hormone insulinase is mainly found in kidneys and liver. It degrades insulin. It splits the disulfide bonds we saw at 7,7 and 19,20 to separate both A and beta and B chains of insulin. Insulin saturates sunbound to carrier protein, and its half-life will be 5 to 18 minutes. Okay, how does insulin work? The insulin receptor is a protein kinase receptor that contains enzyme activity. The insulin receptor is a tetramer having two identical alpha and two beta units. The activity of insulin receptor ultimately produces the following effects. 1. Gene expression in the nucleus of the target cell, leading to biologic action. 2. It will cause translocation of glucose transport proteins to plasma membrane for the insulin-mediated uptake of glucose by the cells. 3. Activation or deactivation of numerous enzymes in glucose and fatty acid metabolism. And 4. Protein synthesis will increase due to increased transcription and translation of enzymes, glucocations, kinase, and phosphofructokinase, pyruvate kinase. This one we know it in glycolysis and others. We want to talk about the tissues and insulin. What occurs? Insulin is required for uptake of glucose by muscles, adipose tissue, leukocytes, and mammary glands. Yes. But the tissues in which glucose transport is not insulin dependent will be, namely, nervous tissue, kidney, RBCs, retina, blood vessels, and intestinal mucosa. These ones do not depend on insulin to take up the glucose. By the other tissues up here, they will depend on insulin in order to take up the glucose. So how does insulin act? It has metabolic effects, it has effects on iron transport, and role in cell growth and development. This is a lot to talk about. Let's continue. Metabolic effects on carbohydrates. One, insulin will increase uptake of glucose in target cells, which are insulin dependent on uptake. Then we have insulin will promote glucose utilization by increasing glycolysis in muscle and liver, or increasing glycogen formation from glucose in muscle and liver by activating the glycogen synthetase enzyme. In glycolysis, we are activating phosphofructokinase. Yes. Insulin decreases glucose production by inhibiting gluconeogenesis, by decreasing uptake of precursor amino acids. It will also inhibit glycogenolysis by decreasing glucose 6-phosphatase levels. Yeah, metabolic effects on lipids. When glycogen concentration increases to around 5 to 6%, glycogenesis is inhibited and additional glucose entering the cells is converted to fat in the liver cells. Insulin also favors hepatic synthesis of cholesterol from acetyl-CoA. The insulin activity promotes deposition of circulating fat into adipose tissue by activating lipoprotein lipase in capillary wall of adipose tissue. Within muscle, insulin suppresses enzyme LPL. It inhibits lipolysis of triglyceride stores. In adipose again, insulin inhibits hormone-sensitive lipase activity, so suppressing lipolysis. Then we have insulin is required for utilization of LDL and VLDLs. We need the insulin to utilize them. Metabolic effects on proteins. Insulin increases transport of many amino acids, especially valine, leucine, isoleucine, tyrosine, and phenylalanine into the cells by increasing membrane permeability. Thus, amino acids are lowered in plasma. Insulin increases the translation of messenger RNA on ribosomes, forming new proteins. In the liver, insulin decreases the rate of gluconeogenesis and thus conserves amino acids for protein synthesis. Effect on iron transport. Insulin increases potassium, phosphate, and magnesium uptake into skeletal muscle cell, and of potassium and phosphate into hepatocytes from ISEF by increasing membrane permeability. Another effect of insulin on the electrolytic balance is to increase reabsorption of potassium, phosphate, and sodium by the tubules of the kidney. Role in cell growth and development. It has an anabolic action which promotes normal growth. Insulin stimulates synthesis of macromolecules in tissues such as cartilage and bone, and thereby directly contributes to body growth. The genes for insulin and its receptors are related to genes that encode a variety of tissue growth factors, which are IGFs, epidermal growth factor, nerve growth factor, and relaxin. And don't forget to like, share, and subscribe. Subscribe for more videos, subscribe for better videos. Yeah, that is it. Thank you. Subscribe. Don't forget to subscribe. Share to your fellow students to also enjoy. Okay, good. Okay.